The story begins in the 1920s with Henri Boulard, a French microbiologist and botanist. While traveling in Southeast Asia, then French Indochina, Boulard observed something curious. Locals were using the skins of lychee and mangosteen fruits to treat diarrhea during cholera outbreaks. Intrigued, he investigated the microbial flora of these fruit peels and isolated a unique yeast strain. This yeast was not the familiar Saccharomyces cerevisiae, the species behind bread, beer, and wine, but a distinct strain with unusual resilience in the human gut. Boulard recognized its potential and patented the strain in 1923. It would later bear his name, Saccharomyces boulardii. At the time, the concept of probiotics was still in its infancy. Elie Mechnikov, the Russian Nobel laureate, had only recently popularized the idea that beneficial microbes could improve health. Boulard's discovery added a fungal twist to this emerging field. For decades, S. boulardii remained a scientific curiosity, overshadowed by bacterial probiotics, but its unique properties gradually attracted attention. Number 1, survival in the gut. Unlike many bacteria, S. boulardii could withstand stomach acid and bile salts reaching the intestines alive. Number 2, antibiotic resilience. Because it is a yeast, antibiotics that kill bacteria do not affect it. This made it especially useful during antibiotic therapy when gut flora is disrupted. Number 3, anti-pathogen activity. Early studies showed that S. boulardii could inhibit harmful microbes including Clostridium difficile, Salmonella, and Escherichia coli. By the mid-20th century, European clinicians were experimenting with S. boulardii as a treatment for diarrhea and gastrointestinal disorders. In 1989, it was officially recognized as a probiotic by the World Health Organization. How does it work? Despite nearly a century of study, S. boulardii still holds mysteries. Unlike bacteria, it does not permanently colonize the gut. Instead, it passes through, exerting temporary effects. Researchers have identified several mechanisms. Number 1, barrier protection. It strengthens the intestinal lining, reducing leaky gut and preventing pathogens from attaching. Number 2, immune modulation. It stimulates the production of secretory IgA, an antibody that defends mucosal surfaces. Number 3, toxin neutralization. It produces enzymes that degrade toxins from C. difficile and E. coli. Number 4, microbiome balance. It helps restore microbial diversity after antibiotics or infections. This multifaceted action explains why S. boulardii is effective across a wide range of gastrointestinal conditions. Historical milestones. Here's a quick timeline of S. boulardii's journey. In 1923, Henri Boulard isolated yeast from lychee mangosteen peels. In the 1950s to 70s, and early clinical use in Europe for diarrhea. In 1980s, commercial production was expanded and marketed as Florestur. In 1989, the World Health Organization recognized S. boulardii as a probiotic. In the 2000s, and widespread clinical trials confirmed the efficacy of S. boulardii in treating antibiotic-associated diarrhea. In the 2010s, and research expanded the use of S. boulardii for treating inflammatory bowel disease, irritable bowel syndrome, and traveler's diarrhea. S. boulardii is one of the most widely used non-bacterial probiotics worldwide. S. boulardii is sold globally as a dietary supplement and prescribed in hospitals for specific conditions. Its applications include: Number 1, antibiotic-associated diarrhea, AAD, multiple meta-analyses confirm its effectiveness in preventing AAD, especially in children. Number 2, Clostridium difficile infections. It reduces recurrence rates when used alongside standard therapy. Number 3, traveler's diarrhea. It helps to prevent gastrointestinal upset in people visiting regions with a high risk. Number 4, irritable bowel syndrome. Some studies suggest symptom relief from IBS, though results are mixed. Number 5, inflammatory bowel disease, IBD. Evidence shows potential benefits in Crohn's disease and ulcerative colitis. Beyond the gut, researchers are exploring its role in metabolic health, immune regulation, and even neurological conditions via the gut-brain axis. The mystery. Why does it work so well? Despite its proven benefits, S. boulardii remains enigmatic. Key questions persist. Why doesn't it colonize? Unlike bacteria, it doesn't establish long-term residence in the gut, yet its effects are significant. Strain specificity. Is S. boulardii truly a separate species or a variant of S. cerevisiae? Genetic studies suggest it is a subspecies, but its probiotic properties are unique. Beyond the gut, could its immune modulating effects extend to systemic diseases? Research is ongoing. This blend of proven efficacy and lingering mystery makes S. boulardii a fascinating subject for microbiologists and clinicians alike. Thanks for watching. Please like, share, and subscribe to the channel Microbe Masters for more insights into the world of microorganisms.